Welcome back, everyone. Hi, I guess I didn't scare you off the first time. I'm Dr. Rebecca Pinko, and this is Basic Plant Science Lecture. This is the second out of 8 lectures, so we move really, really fast through the course of this semester here. So if you joined us for lecture 1, welcome back. Guess you didn't get scared. And if this is your first lecture, make sure you watch the recording of lecture 1 so that you can catch up. So with that being said, I'm going to jump in. This lecture and the next several lectures are going to be pre-recorded. So this is like unique kind of ethos where I'm basically talking into a room with nobody around me, pretending that I'm talking to 35 students. So bear with me a little bit. I'm going to try to be entertaining. I'm going to try to not look at myself in the camera and try to look at you guys, but it's very hard. It's not as easy as it looks. All right, so let's go ahead and get started. I'm going to share the screen just like if we were not pre-recorded, right? Oh, wow, just like real. And I'm going to pull it up here, pull it big. All righty. And so hopefully you guys can see what I am showing you here. Is okay, great. So this, as I said, this is the second lecture. If you need to get in touch with me, here's my email address. I'm very good at getting back pretty quickly, so keep that in mind. Pretty good turnaround time for you guys. All right, great. So let's go ahead. So the last class we reviewed the syllabus. So if you haven't already, make sure you pull that up on Blackboard, Canvas, on Canvas to look at that. And we also introduced the basic introduction to the basic plant science course, right? We had the importance of plants, life on Earth, and then plants and classification. So that's what we did last class. And today we're going to get into a little bit more of the science of it with tissues and stems. So we're going to have a kind of crash course introduction into the plant body and what that is. And then growth. Different types of growth. So plants have 2 different types of growth, primary growth and secondary growth. And then we'll talk about something called meristems and tissues. And lastly, we'll talk about different types of stems, so dicot stems versus monocot stems, and then some examples of variation and adaptation on the stem. All right, so let's do a little intro to plant body, because you guys have seen, like, here's a plant, right? Hello. You guys have seen plants before, but maybe you don't know quite like the names of the plant parts, right? Maybe you know leaf, right? Maybe you know flower, maybe you know stem, but there's other parts as well. So this is kind of like a representative plant, right? This is not a real plant. This is just a representative plant to kind of give you an idea of the parts. So some things, ooh, I'll annotate. Ooh, I love to annotate. I'm not good at annotating, but I enjoy it. All right, so for example, here we have an apical bud, right? At these spaces you see here, this is, so the apical bud we'll talk about in the slides that are coming up. But I just want you to notice some of these regular things that you're used to, right? Like a leaf, right? And we'll talk a lot about leaves when we get to the leaf lecture. But you have leaves, you have shoots, right? You have a root system. This whole top part is a shoot system. So the leaves, stems, and flowers are part of this shoot system. So we will get oriented with a lot of these terms as we go forward in the class. But just to get you an idea, like this is a plant, guys. All right. So we have these different systems, right? So we have a root system, which serves to anchor the plant to the ground. Most of the time it's in soil. Sometimes it's anchored to like a rock or something like that. The root system also absorbs water and minerals. So that's what the root system does. Then you have the shoot system that I mentioned. So the shoot system is your stems and leaves. The stems are going to raise the leaves up toward the sun. So the leaves are those photosynthetic organs where photosynthesis occurs, which we'll talk about in another class. And then we have reproductive structures, AKA usually flowers, attached to the stems as well. And then inside of the root system and the shoot system is something that's called a vascular system. So the vascular system, you can think about it like we as humans have a vascular system, right? We have veins and capillaries and arteries and all of that stuff. So that's our vascular system. But plants have a vascular system as well. And it's composed of two tissues. Xylem tissue is the first one. Here, this is how you say that. Xylem, like xylophone. Xylem tissue. And xylem tissue is going to conduct water and minerals from the roots up through the stem and ultimately to the leaves. And then you have phloem tissue, which is going to conduct basically sugars. So here it says products of photosynthesis from the leaves down through the other parts of the plant. And those products of photosynthesis, which we will talk about in another lecture, are sugars. So you have root system, shoot system, vascular system. All right, so now we'll talk a little bit about growth. Everything grows, right? Living things grow. Plants are no different in that they grow as well. And there's two types of growth. There's primary growth. Let me move this. And remember, I will post these lecture slides as well. So if there's any point where my head like cuts off part of the slide, just know you can pull the slides up on Canvas as well on their own, okay? But yeah, so primary growth is the type of growth that plants do to increase in length, So for example, if you look here on the left, and I'll use red because I have color-coded primary growth with red, right? Look at this little tree on the left here, right? Super short, super tiny, needs to get taller. And the way that it's going to get, well, told you I'm not good at annotating. The way that it's gonna get taller is by primary growth. So it's gonna undergo primary growth to lengthen the shoot system, which we see, and it's also going to be lengthening the root system, which is underground, so we usually can't see that, right? So that's primary growth. When you think of primary growth, I want you to think into your head that it's lengthening. Secondary growth, on the other hand, is to get thick, right? It's to increase the width of a plant body. So primary growth, lengthening, secondary growth, widening. And so that's why I have these little drawings up here, right, of these little arrows to kind of put that in your mind, right? Okay. So all plants do primary growth. Primary growth is something that all plants do, but only some plants have secondary growth. So for example, if we go back to this slide, that kind of makes sense because think about it. Think of like a grass, right? Like a grass. A grass really doesn't need to get thick. A grass just needs to be a grass and grow and do its little grassy thing, but it's never going to need to get thick like this tree that we see here, right? This tree on the right, in order to become a 100-foot plus tall tree, you need to get thicker at the bottom, and your branches need to get thicker because you're holding up so much material, and your roots will need to get thicker because they're supporting this massive tree, right? But for like a grass, you don't really need to get thicker. So grass would only undergo primary growth, whereas like trees undergo secondary growth. I should point out though, so like this thick trunk, right? The reason why that trunk is so thick is because of secondary growth. But you may still at the tippy tip, so like here, at the tips of the branches, you'll still be having primary growth because those branches are still lengthening. So that's why I just want you to put in your head, primary growth, lengthening, secondary growth, widening. And if you have any questions, remember you can write them in Canvas where the slides are posted. You can actually write replies in that, and that's where we can interact as a group. All right, let's move on from here. Yes, sorry. So all plants have primary growth, only some plants have secondary growth, right? Okay, well, who has what? Who has what? So herbs are a type of plant And herbs only have primary growth. They only have that. Herbaceous, the word herbaceous means non-woody, right? So it's always going to be kind of like soft and pliable. So like this time of year, maybe they're finished now, but like tulips are a type of herb. All the culinary herbs that you think about, maybe you have a basil plant at your house, right? That's an herb. Dandelions are an herb. So those only have primary growth. Then you have shrubs. Shrubs are like kind of like small trees, you can think of them that way. But basically they're plants that are bigger. They're bigger plants than an herb, but they're smaller plants than a tree. And shrubs have both primary and secondary growth, and shrubs can produce wood. So for example, here we see a rose bush, right? Maybe bush is something you're more comfortable with, right? If you say that something is a bush, chances are it's a shrub, right? If you have a forsythia bush in your yard or a rose bush, those are shrubs, right? Those are shrubs. And those can do primary and secondary growth. And they can produce wood. And then lastly, trees also have primary and secondary growth, and trees can also produce wood. So one thing you should notice from these 3 slides, look, you're like, oh no, she's annotating again. Herbs only have primary growth, right? And what do they not have? Wood. They don't have wood. So that should put ding ding in your brain that primary growth never results in anything woody. It's always kind of like green and pliable. Whereas the next 2 that we saw here, the shrubs and the trees, they're both capable of doing primary and secondary growth. And because they're able to do secondary growth, they're able to produce wood. So that's one thing you can notice here. All right. So we have the 2 types of growth. How does this type of growth come about? Who's responsible for the growth? And that's what we will talk about a little bit here with meristems and tissues. Okay. This is where it gets a little sciencey, so bear with me, okay? Hold on. There's going to be some bigger words here. Don't get scared. I'll walk you through it. So here is a plant, and at the top you see it says apical meristems lead to primary growth. Okay, so in this drawing here, in this terminal bud right here is what's called an apical meristem. A meristem is what's the best way to say it? A meristem is a type of tissue composed of cells, and those cells can divide into lots and lots and lots of different other types of cells. So for example, maybe you've heard of the term stem cell. Oh, this is a stem cell, and this stem cell can be used to, this stem cell will grow into maybe bone tissue or nerve tissue. Like the stem cell can kind of go whichever way it needs to go. Meristems are that same type of cell where they will divide and produce new types of tissue. And so for the apical meristem, apical means at the tippy tip. So at the, in this case, at the tip top of this shoot. And that's where these meristems are located. And those are going to allow the plant to produce new tissues to lengthen, right? Primary growth is lengthen. And if you look here, we see our arrow again to remind us, right, that we're talking about getting longer. All right. Don't worry about axillary buds right now. Okay. Oh yeah, so because I know how much you love listening to me talk and looking at these slides, but here I'm going to share with you guys a video showing you an example of primary growth. So I'm going to stop sharing this and I'll pause the recording briefly to pull up the video. 1 second. All right, we should be back recording. Hopefully we are. Let me see here. Okay, I'm going to pull this up, share, and I'm going to make it big. All right, so what we see here, guys, this is somebody had planted a sunflower seed in dirt, and this is a time lapse. So this is going over many, many days, right? Many, many days of this sunflower growing from a seed all the way through. But this gives us a good idea of primary growth. So here you go. Here's the new plant. So somebody planted a seed down there, right? So here's the new plant emerging out of the soil. And as it's emerging out of the soil, it's growing new tissues. It's growing new leaf tissues. It's growing new stem tissues. And all of that's happening at that apical meristem. So the apical meristem is dividing and creating new tissues. And some of those tissues are going to be making up the stem. Some of those new tissues are going to be making up leaves. And this is allowing the, in this case, sunflower plant to lengthen and get longer and longer and longer. This is a good example of primary growth. Look, now it has already made many, many leaves. We see at the top there is the beginnings of the actual sunflower. Oh, and now we're at the top looking at the sunflower bud. Right, so that apical bud is capable of making all of these things: leaves, stems, flowers, etc. You see that kind of wiggle that's going on there? We'll learn about that type of movement in the plant physiology lab. Up, there you go. Ah, sunflower, right? Cool. So this is to get you guys thinking comfortably about this idea of primary growth, right? Growing new tissues to allow the plant body to lengthen. Okay, I will pause that. That's basically the end of that. I can, you'll have access to this video via the slides. So let me stop sharing this and go back to our PowerPoint. Oh, geez. Here we go. Okay. Here is where we left off. So primary growth, this is what I was just saying when watching the video. Primary growth at the apical meristems gives rise to primary meristems. So the apical meristems are going to make new tissues. And the first tissues that those apical meristems make are called primary meristems, shown here in purple. So here, what this is going to show you here, we're going to basically zoom in. We're going to zoom into this apical bud, right? Here's apical bud. We're going to zoom in there and look at it up close so that we can see the primary meristems. So here we go. We're going to zoom in. Boom. So we zoomed in. And let me show you where we are, because I love it. So look, here you go again. Apical meristem, right? So that is right here at the tip of this shoot, tip of the shoot. And I just said that this primary meristem is going to make new tissues. And the first tissues it's going to make are something called primary, hold on, I got confused there for a second. Primary, right. Apical meristem is going to make tissues. And the first tissues apical meristem is going to make are these primary meristems, And I will show you where they are. They're right there. So these are the names of the primary meristems that the apical meristem makes. You have the protoderm, which we'll learn about what that makes in slides coming up. You have the procambium, and you have the ground meristem. Those are the three primary meristems, and those are made by the apical meristem. Okay, this is another schematic showing you the exact same thing, but maybe it might make things a little more clear for you. The primary meristems here have also been, well, here. Let me do this. Oh, do I do it by myself? Yeah, okay, I thought maybe I did. So right, so primary growth is occurring at those apical meristems. It's the same thing, just a different schematic. And the primary growth at those apical meristems is going to give rise to these three primary meristems, and there they are again, right? There they are again. In this case, just to start to get you oriented, because this is a little easier to see, The protoderm, so the apical meristem is going to make protoderm, and that's going to be right here. The apical meristem is going to make cambium, which it's hard to see, but it's kind of like right around here. And the apical meristem is going to make ground meristem, right? Another primary meristem, and that's going to be right here. But what I want you to know is primary growth happens at the apical meristem, and that the apical meristem gives rise or makes 3 primary meristems, and these are their names. These are their names. This is from your book. This schematic is from chapter 4. So you feel free to refer to the book. It's got a lot of useful information too. Okay, this is another schematic showing the same thing, just to give you guys, just to hit this home, right? We got the apical meristem here at the tippy top, right here. Right here is the apical meristem. Same over here. On the right is a drawing, on the left is a microscope slide, right? Apical meristem. And that apical meristem is going to give rise to 3 primary meristems: procambium, the protoderm, and the ground meristem, Okay, enough about that. So the protoderm, the procambium, and the ground meristem are still meristems. And so that means that they're able to divide and grow new tissues. And the tissues that those 3 primary meristems make are called primary tissues, right? Primary meristem makes primary tissues. With primary tissues, sometimes you have simple tissues that are only made up of one type of cell. For example, cells with really scary names, so don't get too worried. I'll walk you through it, but you have parenchyma cell, parenchyma cell, cholenchyma cell, and sclerenchyma. It's like fun. I like sclerenchyma the most. So we'll see those on slides coming up. But if you have a tissue made up of one type of cell, we call it a simple tissue. And these are some examples of simple tissues: parenchyma tissue, cholenchyma tissue, and sclerenchyma tissue. However, you could also have complex tissues, which means that they're tissues made up of many different types of cells. And some examples of those complex tissues are, it's not like a complex, like, oh, I have, no, it's like, it's complicated in that it's made up a lot of different types of cells. And some examples of complex tissues are pith, cortex, epidermis, xylem, that we already mentioned before, and phloem, that we already mentioned before as well. Okay. So here's just get you to see what it looks like. So here's simple primary tissues, right? These are tissues made up of one cell type. In this case, we're looking at parenchyma tissue. And even though it's a big scary word, the nice thing is that you see here at the bottom, parenchyma tissue is made up of parenchyma cells. So you don't have to learn another scary word. It's all one word, one scary word at a time. So parenchyma tissue is made up of parenchyma cells, and parenchyma cells are the most abundant cell type found in plants. So you'll find parenchyma cells everywhere in the plant, pretty much. And this is what they look like. Nothing too fancy, you know, pretty round and relatively uniform. Some of these might appear purpler than other ones, but that's just because this is the way that the specimen was cut to be under the microscope slide. This is a microscope slide. So that's parenchyma tissue is one type of simple tissue. Then you have cholenchyma tissue, right? And also nice, cholenchyma tissue is made up of, you guessed it, cholenchyma cells. And cholenchyma cells are really like kind of stretchy. So here it says pliable and strong. But yeah, they're kind of like really long, kind of like stretchy cells that provide this kind of flexible support for growing organs and to mature organs. So for example, if you've ever eaten celery and you've gotten those like celery strings, those strings are actually parenchyma tissue, right? So kind of like long and stretchy and strong. Okay. Then you have sclerenchyma tissue, right? Sclerenchyma tissue. Sclerenchyma tissue is made up of only one cell type, like all the other simple tissues we've just looked at, but there are two types of sclerenchyma cells. There are what are called sclerids and fibers. So on the left here is an example of sclerenchyma tissue made up of sclerids. So sclerenchyma tissue is very thick and it's kind of like has very tough cell walls. And so in the case of sclerids are kind of almost like grit, like little pieces of grit. So if you've ever eaten a pear and you kind of get that like gritty texture that the pear flesh has in it, that's because of sclerids, right? That like little gritty texture is the sclerids. Fibers, on the other hand, they're not gritty, as it were. They're more just like strong and tough. So on the right here, you see a microscope slide, and all of the pink cells are fiber cells. So they're also long. And if you look here on the left, we use fiber cells to make lots of different fibers for our own use as humans, right? So we make fibers out of hemp, out of jute. We even make fibers out of palm and bamboo and other types of plants. So we use it, and also we consume fiber, right? Okay, so those were examples of simple primary tissues. Now I'll show you some examples of complex primary tissues, which means that they're made up of many different types of cells. So one example is xylem, which we have already talked about. Xylem is a cell part of the vascular system in plants. It transports water, right? Transports water from the roots to the leaves, all the way up through the stem. So we're not going to get into great detail with this today, but just suffice it to say that xylem tissue is made up of different types of cells. Sometimes they are tracheid cells, which we see here in A. And then there's also vessel element cells, as we see here in B, 3 different types of vessel element cells. In either case, the job of these cells is to transport water and minerals in the plants, right? But it's more complex than the simple tissues we just looked at. Here's another example of a complex primary tissue, phloem tissue, right? So phloem, as I mentioned earlier, is the part of the vascular system that transports basically food, right? Sugars in plants. And we don't need to know all the stuff here on the slide. Just suffice it to say that phloem is made up of many different types of cells. For example, something called a companion cell, something called a sieve tube, something called a sieve plate. You don't need to know those terms. Just know that phloem is an example of a complex primary tissue. Another complex primary tissue is epidermis. So epidermis should be a relatively familiar term because you have epidermis too, right? Humans have epidermis. It just means kind of like skin. And so on plants, the epidermis is kind of like their skin too. They have epidermis on leaves. They have epidermis on shoots or stems, and they have epidermis on roots. And it serves the same purpose that our epidermis serves. It's a barrier that separates the inside of their body from the outside, right? So it serves to protect and not let things just come in and out. And also in plants, sometimes it secretes like a little thick cuticle, and our skin also has a cuticle. But epidermis is complex in that it's made up of a lot of different types of cells. Oh, and this slide just shows you, this is a cross section through a leaf. And it's just trying to show you that there's an upper epidermis and a lower epidermis. In this slide, the epidermis is this layer of the kind of thicker cells. So this, it's green. And that orangey color above it or in the case of the lower epidermis below it, the orangey thing is that cuticle that I was talking about, which is still part of the epidermis. It's just a different part of it. All right. So yeah, so epidermis is made up of many different cell types. Here's examples of that. So sometimes the epidermis of plants will make little hairs called trichomes. Not always, but sometimes. And sometimes the trichomes are different shapes and have different properties depending on the plant, right? So on the left here, we see these are SEM or scanning electron microscope slides of a plant's epidermis. And on the left here, we see these kind of, this almost looks like aliens, right? This is like some alien stuff. So there's all this like this isn't computer generated. This is a scanning electron microscope slides, but you see the kind of like pokey, scary alien thing? Yeah, so those are trichomes. And over here, this kind of long finger-like thing is also a trichome. Yeah, so you can see here, some of the cells on the epidermis make these hairs or trichomes, and sometimes they secrete oils, and you could see that here, these big purple globs on the left are oil that's being secreted. Likewise, these big, almost mushroom-looking things on the right is oil being secreted from the epidermis. So epidermis, another example of a complex primary tissue. Okay. Alrighty, so we've already learned that you have an apical meristem, and the apical meristem's at the tip, and the apical meristem makes 3 primary meristems, right? There they are in purple: ground meristem, procambium, and protoderm. And each of those primary meristems are going to make tissues that are the primary tissues. Here it says primary growth tissues, but primary tissues is fine. And here are those primary tissues. You have pith and cortex. Those are 2 primary tissues. You have primary xylem and primary phloem. And you have epidermis. So yeah, I was wondering where the rest of this slide was. Okay. That makes more sense. Right. So don't worry about the word sporophyte. That is left over from another class that I teach. And don't worry about vascular. Just worry about primary tissue systems of all plant organs. Just worry about that, okay? So here, you see here, dermal tissue system. So let me get you guys oriented. I'll move this over here. I don't know if it'll move for you guys. So here you got this in A, this thing here, that's a leaf cross section. This thing here in B, this is a stem cross section. And then on the bottom here, C, you have a root cross section, right? And that's why there's a plant here to show you where you are. Look, you're in the root, you're in the stem, you're in the leaf, right? Okay. So you have these tissue systems are in all parts of the plant. So for example, the dermal tissue system, you're going to have it in the leaf, right? So let's use purple. Dermal tissue system, epidermis, right? So that's here on the leaf. It's right here, like we saw on the slide. There'll also be a lower epidermis, upper and lower. This stem shown in B is also going to have an epidermis, right? There it is on the outside, right? The skin. And then this root is also going to have an epidermis, right? This is on the skin. So that's what this is saying here. Dermal tissue system is called an epidermis. Doesn't matter if you're talking about a leaf, a stem, or a root. Always going to make epidermis. Next, we have, I gotta get out of this to be able to, the ground tissue system. So similarly, if you are talking about a stem or a root, the ground tissue is going to be cortex and pith. So let's look at that first. So I'll use yellow. So let's start with stem, right? Here's stem. Ground tissue system, cortex. Whoops, so bad. I am so bad at drawing. Cortex. So all this stuff here is cortex. If you go, oh, and then pith, I guess it's not labeled here. If there was pith would normally be in the middle, but this one doesn't have pith for some reason. But cortex and pith are ground tissue systems in the stem and the root. Similarly, down here in the root, right here is cortex. We'll see pith on other slides. Don't worry the fact that pith's not here. And then the ground tissue system in the leaf is called mesophyll, which we'll learn way more about in the leaf lecture. But what I'm trying to show you with this horrible drawing is dermal tissue system is the skin. So that's on the outside. Ground tissue system is kind of like all the other stuff in the middle, right? As I've drawn very beautifully here in yellow. And lastly, we have the vascular tissue system. And no matter where you are in the plant, if you're in the root or the stem or the leaf, your vascular tissue system for primary tissues is going to be primary xylem and primary phloem, right? So I will use orange. So in the root down here on the bottom, right, the xylem and phloem are located here. I'm not going to distinguish which one is which right now. You don't need to know that. Xylem and phloem in the stem, located right in the middle. Xylem and phloem in the leaf, oops, located in the middle. So I'm just trying to give you a color visualization, right? Dermal system is the skin. Ground tissue system is the middle part, right? All that middle part. And then vascular tissue system is usually restricted to the center of whatever you're talking about. So a root, a stem, or a leaf, you're looking at vascular tissue mostly being localized in the center. Okay, so I know growth is not the funnest thing to talk about, please. It's definitely not my favorite thing to talk about, but it is important for us to establish so that we can like figure out when we're going to the next stages of things. And there's going to be a lot of redundancy here because it's like, don't want you guys to be overwhelmed by these terms. So here we go again. You have an apical meristem, and that's where primary growth occurs. The apical meristem is going to give rise to primary meristems. There are 3 of them: procambium, protoderm, and vascular. Sorry, procambium. Procambium, ground meristem, and protoderm, right? 3 primary meristems. And those primary meristems are going to give rise to the primary tissues. And I tried, probably not successfully, to demonstrate that on the previous slide. So here you go. This is a nice, clean schematic saying all of that much, much more neatly than I'm able to. So apical meristem makes these 3 primary meristems, and each of those primary meristems is going to make primary tissues. There you go. Sometimes words are better. Look, and I even color coded it for you. You're welcome. All right, that's all been primary growth so far. That's all we've talked about so far is primary growth. Now we're going to jump into secondary growth. I'm going to go refresh my tea, and let me stop this real quick. I'm going to go refresh my tea and I will be right back. Okay, I'm back. Tea is refreshed. We'll keep going. All righty. Oh, geez. Yes. Okay. So we left off here. Yes, right here. Right, so far we've only talked about primary growth. Now we're going to talk about secondary growth. Remember, all plants do primary growth, but only some plants do secondary growth. For example, shrubs and trees. So secondary growth is by lateral meristems. As primary growth was from the apical meristem, secondary growth is by lateral meristems. Because remember, the purpose of secondary growth is to get wider. And there are 2 lateral meristems the vascular cambium and the cork cambium. Both of them are shaped like a ring. And the vascular cambium is going to give rise to secondary xylem and secondary phloem in roots and stems. The cork cambium is going to give rise to cork, also known as phellem. Cork is fine. And phylloderm in the roots and stems. And you might notice that leaves are absent here, right? No secondary growth in leaves. That's because leaves only do primary growth, right? You never have you'll never have a woody leaf. Leaves just need to get long, they don't need to get thick. Yeah, there's probably some exceptions out there because there's so many plants, but yeah. And this little arrow right at the bottom is just reminding you secondary growth is by lateral meristems because you're trying to get thick, right? Wider. Okay, so here is a nice schematic that shows you all of the players of primary and secondary growth, right? That apical meristem makes 3 primary meristems. Those primary meristems are going to make primary tissues. And it's kind of like, all right, and there are primary meristems and secondary meristems, or AKA lateral meristems that I just mentioned on the previous slide. Oh, it's hard to talk so much in a room all by yourself. But where do these lateral meristems come from, right? If you look here, you can kind of see an inkling of it. We know that the primary meristems came from the apical meristem, but where the heck do these lateral or secondary meristems come from? They're actually produced by some of the primary tissues. You don't need to know which ones, but I just want you to know it didn't just like whammo show up. It is actually made. So at a certain point in the stem or roots development, at a certain point, the cortex will undergo changes and make the cork cambium. Similarly, at a certain point in the stem or the roots development, there's going to be, and again, you don't need to know the names, but the vascular cambium is going to be made from some of the ground tissues and also some of the vascular tissues. You don't need to know the names. I'm just showing you that the lateral meristems come about because of chemical changes that the primary tissues undergo. That's how they come about. Finally, when you do have the lateral meristems, right, the cork cambium and the vascular cambium, they're going to make tissues themselves because they're meristems. Cork cambium, as I said on the previous, is going to make cork and phylloderm, and vascular cambium is going to make secondary xylem and secondary phloem. Because remember, though, secondary growth, widening, right? Don't worry about periderm. I won't ask you about periderm. Let's just put it that way. All right, here's another overview of primary and secondary growth, but in like a visual way instead of just the text, right? This is all text. Let's like look at it kind of more in a visual way. This is the same information that we just covered. So here you go. You're at an apical meristem at the tip. The apical meristem is going to make those primary meristems, right? Protoderm, procambium, and ground tissue. Ground meristem. And those are going to make those primary tissues, right? You could see them there. Epidermis, cortex, primary phloem, primary xylem, and pith, right? So that's your primary growth. Notice it's occurring where? At the tip of the plant, right? Because we're lengthening for primary growth. Okay. But down here on the bottom part of this plant, in the stem, we're starting to see the secondary growth, right? And the secondary growth comes about from these lateral or secondary meristems, cork cambium and vascular cambium. And those meristems are going to give rise to secondary tissues, which we can start to see some of those here. For example, secondary phloem and secondary xylem. Notice you still see on this slide notice you still see like some of this, like you still see primary phloem, you still see primary xylem and pith and cortex, which those are all primary tissues. The reason why you're seeing a combination of primary and secondary tissues in one part of the stem is because in order to go from primary growth to secondary growth, you're going to have a transition. It's not going to be like the stem's doing primary growth and then just making secondary growth. Remember, I said that the secondary meristems come about from the primary tissues. So depending on what age you're cutting that stem, you might see a combination of the two. That'll become clearer, I think, on the following slides. Let me clear that. Okay. Yep, and there's our arrows again, right? Right, because primary growth is for lengthening and secondary growth is for widening. All right, this is the same thing, but kind of walks you through it step by step. So here's our ground. Here's our ground. Here is our primary meristems, right? Here's our primary meristems. They give rise to these primary tissues: pith, cortex, primary phloem, primary xylem, and epidermis. Eventually, some of those primary tissues will give rise to secondary or lateral meristems. Here, we're starting to see one of those, right? Oops. Vascular cambium shows up. Vascular cambium makes secondary xylem and secondary phloem. So we're starting to see those secondary structures. And then eventually cork cambium shows up, which isn't shown on this slide. It's just saying periderm. Don't worry about that. Just the point is that you see these slides here show you the transition. This is all primary growth here. But then eventually when the stem needs to get wider, we switch to secondary growth to allow it to get wider. Okay. Eventually there will be no primary tissues remaining in the stem, only secondary tissue. That's what I'm trying to get at here. Okay. So we learned there's the vascular cambium and the cork cambium, right? So look here, cell of vascular cambium. So this is showing you how, remember, the vascular cambium and cork cambium are shaped like a ring. And so it's not, you can't see the whole ring here, but you could picture it going like something like this, right? and off the slide. But this is just trying to show you that the vascular cambium makes secondary xylem toward the outside of the stem. And it makes, the vascular cambium makes secondary phloem to the outside of the stem, and the vascular cambium makes secondary xylem toward the inside of the stem. Hold on one second. Okay, sorry about that. I accidentally pressed pause. Here, let me reshare. Oops. Grab this. This is why recording is better sometimes, because you can fast forward through these little floops. Okay, back to this. All right, yeah, so this is just trying to show you the vascular cambium makes secondary xylem towards the outside, towards the inside of the stem. I don't know why I keep doing that. And secondary phloem towards the outside of the stem. Yeah, you can see it here on this slide. All right, this is a cross section through a stem, a real life stem. See, this is a drawing. This is a real life stem. And I just want to show you where the stuff is. So first we look for the vascular cambium, which is right here, and it's pointing to this right here. That is the vascular cambium. Remember, vascular cambium is a lateral meristem, right? Secondary meristem. And I'll use pink. So look, phloem conducting cells. So the vascular cambium is going to make phloem toward the outside of the stem, right? So like this way. And I'll use light blue. Where is our, here we go, xylem. So the vascular cambium is going to make xylem or secondary xylem toward the inside of the stem. Toward the inside of the stem. Okay. All right, enough of that. Growth is a little tricky. It's a lot of cells and tissues and things like that. I know it's not probably the funnest. Just hang in there. Yeah, it's something we have to discuss so that things make sense later when we're talking about other stuff. Okay, so let's talk about mature stem. So, and by mature, I'm talking about like twigs and branches and trunks. Twigs and branches and trunks. So here is a twig, right, or a small branch, and some of the players that we're used to seeing, right? Here's our terminal bud. That's where the apical meristem is going to be. Here, so this one in A, it's the same thing as is shown in B, except A is what it would look like in the winter time when the leaves fall off, and B looks like how it looks now, right, in the spring, summer time where there's leaves on it. A few terms, since we are talking about stems today, is this term node. So node is a region on a twig. It's a region or a branch, twig or branch. It's a region. So like this region is a node. This is another node. Here's another node. On this one, here's a node, here's a node, here's a node, etc. Look, this is a node, node. So it's a region. And the reason I point out this region is because things occur at that region. So for example, here at the node, we see axillary buds. Look, there's an axillary bud right there. At the node, we also see leaves, right? So here you go again. There's an axillary bud right there, and there's a leaf. Here's an axillary bud right here, and there's a leaf. So these structures, axillary buds and leaves, occur at the region known as a node. Okay? Hold on. There's a reason I'm telling you this. Then you have this other thing here. I guess I'll use, running out of colors, I will use yellow. You have this other word, internode. Inter means between. So if node is a region, internode is the region between the nodes. So there's an internode, there's an internode, whoops, there's an internode, right? Likewise, this is an internode, this is an internode, etc. The reason why I'm telling you this is because you can tell how old a twig / branch is by counting the nodes. Yes, that's why I'm telling you that. And that's kind of cool because let's say, for example, let's say you're walking somewhere like in a park or something, or your friend's backyard, and you see a bush that you were like, that's a very beautiful bush. I'd like to put that in my backyard. You might want to think like, well, how long will it take if you buy that bush, a bush like that? How long is it going to take for that bush to get to the size of your friend's plant, right? So you can start looking at the twig and you can start to count the nodes. And you could say to yourself, well, look at this, like this here. Oh, I kind of screwed it up. That's okay. Let's just go with node for now. But like, you can tell how old it is. Yeah. By count. I screwed it up. Oh, so close. So close. This is going to be annoying. Okay. Those nodes and internodes, yes, those are important things. Nodes and internodes are important, but sorry, you can tell how old a twig or a branch is by counting these things. My bad. Bud scale scars. Sorry. So for example, node and internode is still important. when we go into other lectures, we're going to come across node and internode again. So there's still, and it's important to know, like you can say like, oh, look, where is the leaf? The leaf and the axillary bud are at the node. Fine. But that's not how you tell how old a twig is. You tell how old a twig is from these things called bud scale scars. So remember how I said this one in A is like the wintertime one, and this one in B is like the springtime one, or spring summer one? Here's how you think about it. So here's this terminal bud. When spring happens, the terminal bud's going to fall off, and primary growth is going to, sorry, the outside of the terminal bud is going to open and fall. This little scale that covers it's going to fall off, and that's going to have expose the apical meristem, which is going to allow primary growth to occur. And so that's what would have happened here. At one point, there would have been a terminal bud there. And then when spring came, it would have woken up and grown to that. So I kind of did that in reverse. The point is, you could tell how old a twig is if you count these things called bud scale scars, because that's where the terminal bud was. So the terminal bud was here. Whoops. Terminal bud was here. And then spring came and the plant grew, right? Like this. It grew. And so this section from here to here is 1 year old. So 1 year old. If this were to grow, then that would be a second year. Oy, this derailed very quickly. This is not want to spend this much time on there. Okay, trunks. Trunks. Woody. So we're talking about secondary growth. Look at these tree trunks, very wide, right? Very wide, so lots of secondary growth. Look, this is a person and standing in front of a tree. This tree has abundant secondary growth. Look how wide it is, right? Look how wide it is. What made it wide? These lateral meristems made it wide, right? Because they were able to grow those secondary tissues, secondary xylem, secondary phloem, cork, and phyloderm. Here is a cross section through a trunk, and you can see secondary tissues when you're looking at a slice of a trunk. Here's a microscope slide showing you a slice through a trunk, a very small trunk, because this has to fit on a microscope slide, right? Here is the same exact thing. This is an actual picture, and this is a drawing on top of that picture to show you some of these things. So you don't have to know everything on this drawing, but I'll point out things we've covered so far. First, look for the two lateral meristems, vascular cambium and cork cambium. I've already said it, but we'll say it again, that the vascular cambium makes secondary xylem toward the, toward the inside, right? And secondary phloem toward the outside of the vascular cambium. Let me move this thing. And the cork cambium up here makes phyloderm toward the inside of the stem and cork toward the outside of the stem. One thing I want you to notice on this, though, is I don't know if you've ever heard of like tree rings, like, oh, you can tell how old a tree is by counting the rings. Every year, the vascular cambium makes secondary xylem and secondary phloem every year. But because the vascular cambium is like growing wider, it will crush the secondary phloem over time. So secondary phloem can't build up. But because it's getting wider, the secondary xylem that it's leaving inside can remain. And so, for example, here, look, see this line? So this, between here and here, this is 1 year's worth of secondary xylem or wood. This is the previous years right here. This will become clearer on other slides. I'm just pointing it here since it's already labeled. Okay, this is a much simpler schematic compared, this is a more complicated one, right? So this is a much more simpler one, show you the same thing. But first we look for those lateral meristem, cork cambium and the vascular cambium. Cork cambium is going to produce cork to the outside and phelloderm toward the inside, right? And yes, all three of those tissues make up the periderm, but I don't really care about you guys knowing periderm. Vascular cambium is going to produce secondary phloem toward the outside and secondary xylem toward the inside of the stem, right? This is what I was just trying to draw, right? So the secondary xylem is going to grow one ring every year. And each ring has what's known as early or late wood. I'll show you here. Okay. Do you notice how like here, these cells are much smaller, much more dense compared to like these cells up here or these cells down here? That's your early wood, late wood type thing. You can see here, one ring has both early wood or spring wood and late wood or summer wood. That's because every ring of secondary xylem that's made is made over the course of a growing season, right? Over the course of a year. So in the spring, there's a lot of water and rain. And so the cells are going to be growing much faster. And then so they're going to be bigger. Hence this kind of wider circle of these cells. Then the summer comes, it's super hot, doesn't rain as much. And so the secondary xylem cells that are made in the summer are going to be smaller in size. So this is your spring wood, this is your summer wood, and this line here would be your annual ring. Whoops, okay. This line here would be your annual ring, right? Composed of both spring and summer wood. And then the next year, look, we have spring wood up here, but we don't have any summer wood yet. I probably draw this on it. Yep, there you go. Annual ring. And look, so this is what's cool about annual rings is that you can count how old a tree trunk is by counting the annual rings. So I'm not going to count them all here, but at least show you where they are, right? So like each of these circle thingies, I can't draw, right? 2, 3, 4, etc. Those are all annual rings, each one composed of both spring and summer wood. Okay. Okay. Wood rays. Well, I mentioned parenchyma cells earlier about how they're the most abundant cell. Wood rays are just, I don't care. Here, this is a better way to show you. Wood rays run through the secondary xylem. See? And if you look at this cross section, what did I use? Pink? Wood rays, pink. Yeah. Look. Wood rays radiate out from the center. So here you go. See all these. So you see the annual rings on this tree trunk, right? But then you might also see these long kind of lines radiating out of the center, right? Almost like spokes of a bicycle wheel, right? Radiating out. Those are the wood rays. And what those do is they help move different things from the inside of the stem to the outside of the stem. So for example, water, some of them will help move water that way, and some of them will help move food that way. Just so you know. Oops. All right. Once you get into a huge giant trunk, like the one I showed you of that person standing in front of that big trunk, if you were to cut down that tree and look at that trunk, pretty much everything inside of that trunk is just going to be secondary xylem at that point, aka wood. So if you have wooden floors at your house, or a wooden table, or wooden doors, or wooden chairs, all of those things are actually secondary xylem. So you could say I have secondary xylem floors, or secondary xylem chair. But what we call, what you and me call wood is technically a secondary xylem, and this is a close-up, another scanning electron microscope showing you a close-up of secondary xylem cells in a big mature stem. The reason I wanted to show you this is because it really gives you a better picture of those annual rings. I'll use black. So for example, if I showed you this microscope slide and I said, how old is this tree, right? How old is this tree when it was cut down? You'd have to look for the annual rings to make a good guess. And when you count the annual rings, you're only counting the annual rings of the secondary xylem, not all the things that are in a ring shape. So secondary xylem is the stuff that's located right here. Oh, oops. All this here, secondary xylem. And if we look, 1 ring, 2 rings, 3 rings, right? And there's pith located here in the middle. The point is here, right? 1 ring, 2 ring, 3 ring. We know this tree is at least 3 years old, right? Because we see 3 rings of secondary xylem. In all likelihood, it's much, much older than that because it takes a long time for most trees to get old enough to even make the secondary structures. But we could say at least 3 years old at the very least. Don't worry about resin canals. I don't care about those. And I don't really care about this part either. I just wanted to show you, if any of you are carpenters or do any sort of stuff with like wood and you care about wood grain, right? Wood, like the grain of this wood. I just want to show you that a block of wood will show you different structures depending on how you cut it. I guess if you're a carpenter, you probably already knew that, but maybe you're an amateur carpenter, right? Maybe you wanted to start, maybe during the pandemic, you were like, I'm going to start building things. So this could help you, I guess. But yeah, depending on how you cut a block of wood, you'll see different structures. So for example, this is an easier one to kind of wrap your head around. But here's like a cross section. So if this is a block of wood, and if you did a cross section cut is like this. This is a cross section cut, right? You cut the tree down. You're just looking at the tree cookie, as it were. If you were to do that, these are the types of structures you would see, right? You got your, let's just show you something that you know already, but like, for example, early wood, right? So like all this here is your secondary xylem early wood. Don't worry about that. I'm just trying to show you that what you'll see when you cut it will look different ways. So like that's what it looks like if you do a cross section. If you do what's known as my printer just woke up for some reason. It was really weird. Must be updating. Maybe I'll pause while it does that. Nah, let it do its thing. It was like, yes, tangential section. I don't know if you heard that through the microphone. Yeah, so tangential section looks different. Whoops, and radial section looks different. Main reason that the radial section looks different is that you can see a wood ray radiating across. Yes, okay, right. This is showing you the same thing, right? So like the top here is yeah. Here is, sorry, I'm just getting oriented, radial, and yeah. So here is your, just so you can see it on a real block of wood. This is your cross section. This is your tangential, and this is your radial one. Although for what it's worth, I don't see the wood rays so easily in this drawing. Yeah. This is just to show you that different types of trees have different looks to them in terms of the anatomy of the secondary xylem. So some secondary xylem stays, the rings are very close together. For example, I'll use a different color. For example, here in this bald cypress, you can see how close the secondary xylem rings are together. So they're a lot harder to count. than say up here. Another thing you can see here, look, white oak, the wood rays are really visible on the white oak, whereas on this red spruce, I don't see any wood rays. Yeah, so just to give you a sense of like the diversity of what different types of wood look like, depending on the tree. This is a nice little slide too, because it kind of gives you a little history of like what each type of wood is traditionally used for and why. But you don't need to know anything from this slide. I just wanted to show you it for you to appreciate. I think we're almost done here. One other thing I wanted to mention is heartwood and sapwood. So this is a cross section through a tree, right? This is a cross section through a tree. And all this stuff in the middle here, so the bark is on the outside here, but like all the stuff in the middle is secondary xylem. However, think about if you have a tree that's like 4 or 5 feet wide, all the secondary xylem doesn't need to be functioning at that point. In fact, it's probably not useful because it would be like having a straw that's like 3 feet wide. Doesn't really work so well. So when a tree gets to be that big, the secondary xylem toward the inside of the tree becomes non-functioning. It's basically like scaffolding. Whereas the secondary xylem toward the outside of this tree still functions in transporting water. So the secondary xylem that still functions is called sapwood. Let me get rid of this thing here. Sapwood. And on this tree, the sapwood is this lighter color here. And the darker color on this tree is the heartwood, and the heartwood would be secondary xylem that is no longer functioning. It just serves as support and doesn't transport water anymore. The reason I like to include this, there's 2 reasons. Number one, again, if you're into certain types of woodworking, see here like this black walnut, see how it's darker toward the inside and lighter toward the outside? That's because this darker section is the heartwood, which is non-functional anymore. And the lighter part is the sapwood, right? This one too, and the white oak, you can see heartwood versus sapwood on that. But the reason why I'm telling you that is because if you're ever like, some people like to graffiti on trees or like scratch into trees and things like that. I'm pointing this out because the living part of the tree that is transporting water for some of these really big trees is sometimes only about 1 inch below the bark. So you might think you're not making a big difference in that tree's life by, you know, carving something into it, or maybe a fence is kind of, let's say a fence is growing through it and it's kind of like this, because you're like, well, look, all this other part of the tree is not even being affected. But all this center part is not even functional anymore. It's just this part here at this point. So that's another reason I like to mention that, is that sometimes for very, very, very big trees, it's only about 1 inch deep into the tree that is still functioning with the xylem. Okay. There you go. There's me. There's me in Queens, showing you this was a ash that had been cut down due to the emerald ash borer, which I don't know if you've heard of that, but it's a very problematic insect that's killing ash trees. So this was cut down about 2 years ago, I think. But look at how big this tree was. Look at all of that secondary xylem inside, right? Or AKA wood, right? It's basically everything in here at this point. Everything in here is wood, right? Or secondary xylem at this point. Okay, I made my husband take that. Look at this tree. This tree also has a lot of secondary xylem, right? Abundant secondary xylem. And this is kind of what I was getting at before, is like the majority of the inside of this tree is non-functional. So it doesn't like affect it too much to have cars drive through it because it's only the outside that is functional. Now, granted, you had to remove outside in order to make a big hole in it, but just found this funny picture. Okay, we're almost done. Let me look. Stems of monocots and dicots. Okay, we got 9 slides left. It's another reason why it's good to pre-record these, because you guys can take copious breaks if needed. All right, stems of monocots and dicots. What are dicots? Dicots are some plants. So in all the plants that exist, about 200,000 of the plants that we know of today are what are known as dicots. So some examples of dicots include cacti, this pretty thing called an anemone, California poppy, but there's a ton of other, pretty much most of the plants you're familiar with are probably a dicot. There's trees, shrubs, herbs, etc. Then you have the monocots, which still have a lot of species, 90,000. And things in the monocots are grasses, lilies, orchids, palms, bananas, etc. But monocots and dicots are different in many different ways, which we'll cover in the flower lecture. But since today we're talking about tissues and stems, I have to point out the fact that dicots and monocots have different arrangement of their vascular tissue in their stems. And so you can tell them apart based on this. So on the left, you see dicot stem cross-section, and on the right, you see monocot stem cross-section. In the dicot stem, I'm going to use red just for visualization, but in the dicot stem, vascular bundles are arranged in a ring. So look, here's vascular bundle, here's vascular bundle, here's vascular bundle, etc. They go all the way around. Vascular bundles are bundles of xylem and phloem, with xylem pointing toward the inside of the stem and phloem pointing toward the outside of the stem. But either way, in the dicots, the vascular bundles are arranged in a ring. In monocots, the vascular bundles are not arranged in a ring. They are scattered all around and random. So like each of these dots is a vascular bundle. And as you can see, they're not arranged in a ring, right? They're kind of all over the place. So that's how you can tell them apart when you're looking at their stems. Here's the same thing, but a close-up. So this is a close-up of the monocot stem. So notice on the left, the vascular bundles are not arranged in a ring. And if you were to do a close-up on one of these guys, you can see all of the different components of it. I always think these look like faces, like, ah. And if you didn't think that before, now you always will. So that's another way you can kind of, if you're trying to tell a monocot stem apart from a dicot stem, I always think like the monocot stem has vascular bundles that are scattered, but also that the vascular bundles look like little faces. Ah. So that could help you. Whereas the dicot vascular bundles do not look like faces. So here you go, it's a dicot stem. Vascular bundles are arranged in a ring, right? In a ring, here's the ring. And if we were to do a close up of one of these vascular bundles, right, that's what you see here on the right. Another thing, so monocots have vascular bundles scattered in terms of the stem. Dicots have their vascular bundles in a ring. Another thing is that you see here, there was no vascular cambium here. Nope, none. And there is one here, and that's because dicots usually do undergo secondary growth. So on the left, you see an example of a dicot. They usually do undergo secondary growth at some point in their life, whereas monocots rarely do, right? So orchids don't get very woody. Grass, as I mentioned, doesn't get woody. This here is showing, I think, sugarcane or bamboo. I think sugarcane doesn't get very woody, right? So another way to tell them apart. This is the last part of this lecture, yay! Okay. Modified stems. So here you go. Sometimes because of different evolutionary pressures, certain plants have adapted to their environments by having these kind of stem modifications. A lot of times the purpose of these stem modifications is to store food or water. So in the case of like this tuber, This here is a stem that's storing food for the plant. You can see here, this cactus is an example of a stem that's storing water for the plant. Here's another example of something storing food. So this is something called a corm. And look, the stem is all big and wide because it's storing food for the plant, as opposed to a bulb where the wide part is actually leaves and the stem part is just this little tiny thing. So like if you've ever cut a onion, it's just that little weird nubby thing at the bottom, that's the stem. All the other things are actually modified leaves. And in other situations, the variation on the stem is to help the plant either climb or kind of spread out. So for example, here with a stem tendril, tendrils will help the plant climb, whereas this rhizome and runner will help the plant grow to new locations. All right, that took longer than I was expecting. So a quiz of this material is on Canvas now, and it will be due by Wednesday at 6 p.m. And Wednesday will be our next class, and then we're basically over a third through the semester, which is crazy. And we will be talking about the leaf and the root next time. So not as boring as STEM, I promise, and not as much tissues too, I promise. So thanks for hanging in there, and I will see you guys next time.